Hello students, how are you all? I hope so you all are fine. Myself Nilofer Khan from GTU University. So previously we have completed the first half of the biotechnology chapter. Okay. So now biotechnology and its application. So now we are going to study the third and the fourth subtopic of biotechnology and its application. So okay, now we are going to deal with the third topic, third concept, that is transgenic animals. So what are transgenic animals? Can anyone say me what is transgenic animals? So the animals that have the DNA manipulated, okay, their DNA is manipulated. We are putting some foreign gene in their body, okay. So those animals are known as transgenic animals. Okay, so moving on to our chapter. So in this chapter, now we are going to study why these transgenic animals are important. What are they doing to humans? How are they beneficial for humans? Okay. So these all things we are going to study in this. Is it clear? Okay. So transgenic animals, as I said, they have their DNA modified. Okay. What is the main thing? They have their DNA modified. You need to remember the points, what all I will be writing down. Is it clear? So their DNA will be modified. Why their DNA will be modified? To possess and express an foreign gene. Okay. So nowadays there are many diseases are there, many drugs are there. So first, before giving that drug to the human being, we need to test that drug. Is it or not? So in this, so today, like we can't test the drug on a human. I can't say to someone that please come here, I want to test something on your body. No one will be doing that. Okay, we are not putting our life to a risk. So what scientists and all are doing, they are taking some transgenic animals, and in those animals they are injecting this foreign gene, and they are noting down the impact. They are noting down the changes, what all is happening in those animals. Is it clear? So those animals are called as transgenic animals. So what are the examples of transgenic animals? Means there are many transgenic animals. Like you can say rats, mice, sheep, and what else? Can you say any two? Any two can you name, students? Pigs are also transgenic animals. Sheep, mice, rat, pig, cow, fish, etc. So these are some transgenic animals. On those we can specifically design, okay, to study their gene regulation, to study their impact, to study their effect. Okay, what all the changes in their normal body functions that we can note down. Okay, so mice, what is this mice? You can see this one, mice. So majority, you can say approximately like 95% transgenic, whatever we are doing, transgenic animals are mice. Okay, and mice is the first transgenic animal. Okay, whatever we are testing in today, everything will be done on mice. It is constituting 95% of the transgenic animals. So this is about the transgenic animal. I hope so transgenic animals are clear. Now we will moving out to the reasons why we are using these transgenic animals. The first reason, the basic, the common reason, just now what I said you, animals will not allow. Is it clear? If they are testing some drug of diabetes, if they are asking me, ma'am, we want to test diabetes drugs on you, whether I will agree? No. I will say, no, I can't do that because I don't know the consequences, what it going to happen. Is it clear or not? So that's why the major thing why transgenic animals are used is this. Then now we are studying each and every point. So first point is normal physiology and development. Okay, first point, normal physiology and development. So in this, transgenic animals, they can be specifically designed to study their gene regulation. Okay, after using a transgenic animal, we can check or we can study how the impact is there on their body, how the gene regulation is there on their body. Is it clear? So how they are affecting their normal body function, how they are affecting their development in their body. Okay, so normal physiology and normal development of the transgenic animals can be studied using these transgenic animals. Is it clear? So one example I'll be giving you. Here it is given, see, insulin like growth factor. Okay, so insulin like growth factor. So what does this insulin like growth factor means? It is one example which is given in your NCERT. So the study of this gene, this factor, that is insulin like growth factor. So we can inject the gene. We can inject the gene in the transgenic animal, and then we can study the changes which is going to happen, and we can see the biological effect what it is going to happen. Is it clear? Understood what is the normal physiology? The word itself, it determines the normal physiology of the body, the normal development of the body can be noted down, can be studied using this transgenic animals. Is it clear? Next, second one, study of diseases. So nowadays you can see there are many diseases coming out. Okay, new diseases are going to be developing today on. Okay, so these transgenic animals are being used as a models here. Just now I said na, human being, they cannot be a model to anyone. Okay, so that's why these transgenic animals are a model. Human beings are using them as a model. They are testing them by inserting the foreign gene. Is it clear? So transgenic animals are also very helpful to understand how gene contribute to development of the diseases. Is it clear? So many transgenic animals are created to enhance our understanding of how genes contribute to disease development. Just now I said this point. Is it clear? So today these are playing very important role because I said many diseases are emerging out nowadays. So for knowing this many different human diseases, they have given some diseases also. I'll show you. You can see this one. Okay, no problem. I'll say you. So there are many diseases nowadays. So these transgenic animals are playing a very important role in knowing some human diseases. Which are the human diseases? Examples they have given in your NCERT, that is cancer. Cancer, you know, it is a very deadly disease. So cancer, if we want to test on a human, no human being will be ready. Is it clear or not? No human being will be ready to become a transgenic for the disease cancer. So these transgenic animals are helping out to human beings. They are acting as a model for human beings. So why? To know the diseases, to know the impacts of the diseases, to know the cure of the diseases. So in your NCERT, they have given you some example, that is cancer, then cystic fibroids, then cystic fibrosis, then arthritis. You know all these diseases, rheumatoid arthritis it is, then Alzheimer, ZH. Okay, so for knowing these all diseases, these transgenic animals are playing a very important role. Is it clear? So first point, second point, I hope so everyone you understood these 2 points. In first point, nothing we discussed. We just understood how the normal physiology is changed. We will be noting down by using a transgenic animal. And in second point, we have studied Nowadays many diseases are emerging out, so for testing those diseases, for knowing the impact, for knowing the cause, and for knowing the what will be the drugs which will be used for that disease, for that one we are using this transgenic animals. Okay, so next. Okay, so next we are going to study the third point, that is biological products. So biological products means what? So for the medical treatment, we require the biological product. Is it okay? So the production of the biological product, we know it will be very expensive. Is it clear? So certain medical treatment is require some biological product, and that products are very expensive. We know that. So transgenic animals, they are generated by introducing some specific gene, some specific DNA segments we are introducing in their body, so that they will be giving us some biological products which will be very useful for the human treatment. Understood? So biological products, we know they are very expensive, so transgenic animals are used to produce those products. In short, I am explaining you. Okay, so here also for the purpose of biological products, we are using these transgenic animals. Okay, so in humans, the examples I will be giving you, I'll write it here. Whatever I will be noting down, those will be the important points from that topic. So you also should pay attention of what I am writing. Is it clear, students? So example I will be giving, human protein. Yeah, here it is, you see. Transgenic animals are generated by introducing specific DNA segments. These products such as human protein, that example is given alpha-1 antitrypsin. That is alpha-1 antitrypsin. So this alpha-1 antitrypsin, this is used to treat the disease that is emphysema. Emphysema is the disease which is used to treat, is a disease which is treated by this protein, that is a human protein alpha-1 antitrypsin, and this protein is produced by a transgenic animal. Okay, we are getting this from transgenic animal. It is a biological product which is used to cure this disease, and this protein we are getting from a transgenic animal. Not only this disease, treatment of PKU, that is phenylketonuria. Phenylketonuria. So this is also a disease which is, and one more thing they have given, cystic fibrosis. Another one, cystic fibrosis. Okay, so phenylketonuria and cystic fibrosis is the diseases which are are the byproducts, the biological products we are getting from a transgenic animal. Is it clear? So one more example is here, rosy. So they have given the rosy about the cow. So what is this? I will be explaining you. So you can see here, in 1997 what was happening. In 1997 what they did, they have taken, okay, before this I'll be saying you, infants are there, small babies and all, why don't we give them cow's milk? I think everyone know them. Cow's milk, what it is having? Cow milk is having less protein. Cow milk will be having less protein and more fat, we all know this. Then human, what they are having, human milk is having low fat, vice versa, low fat and high protein. Okay, so cow they are having less protein, more fat. Humans, they are having more protein, less fat. Okay, so for a baby, for an infant, for a development of infant or babies, both are necessary, as well as good fat is also necessary, as well as protein is also necessary. So what the people in 1997, this has happened in 1997, so you have to remember the dates also. So in 1997, the people have studied this thing and they have introduced one technology, this one only, transgenic animal. So what they did, they have inserted a human gene in a cow, and then they have got a first transgenic cow in 1997. Okay, when they are inculcating a human gene, so human is having high protein, so that cow will be producing what? High protein. Understood everyone? So already it is producing more fat. Now when we are injecting, when we are making that cow a transgenic cow, then it will start producing more protein also. So more protein, more fat. Now you understood how they transgen. Is it clear everyone? You see here. Okay, so in 1997 there was a first transgenic cow that was named as, what I said, Rosie. They named it Rosie. In 1997, you know the names Rosie, Rani, and all. Okay, so that's why you will be getting this type of names. So in 1997, there was the first transgenic cow that was produced human protein enriched milk. Okay, so the quantity I will be writing here, 2.4 gram per liter. This is the protein content. So this milk will be having 2.4 gram protein per liter in their milk. Okay, so the milk will be having human alpha. What is the protein name? I'll write here. This is extra what I am saying. Okay, alpha lactalbumin. Right. Human alpha lactalbumin. So this is the human protein. Okay, so this protein, it's nutritionally very good. The milk which we are getting from Rosie, it is nutritionally very good for babies and infants. Is it clear now? Now you are clear, I think, how we are getting the biological product from the transgenic animal. This was the one example which I have said. There are many other examples how the transgenic animals are giving us biological products. Is it clear now? Next. Fifth point is chemical safety testing, toxicity testing, and safety testing. So this chemical safety testing is also nothing but, wait, here one more point is there, that is vaccine safety. First I'll complete this. So nowadays we know coronavirus vaccine is very famous, everyone know that. Okay, so many vaccines are there, so that vaccines are not directly given to the humans. Before giving it to a human, that vaccine will be tested on some transgenic animal. So for this purpose, for not this purpose, for many 95% cases we are using a mice as a transgenic animal. Is it clear? So here we are testing the vaccine on the transgenic mice before using it on a human. Is it clear? Polio vaccine is there. Polio vaccine also we test on mice. So many vaccines are there. They have not given a particular name. So if the vaccine is successful on that transgenic animal, then only they will be giving it to a human. If it is not successful, means again they will be working on that vaccine. They will improve it again. Is it clear? So first what they are doing, they will make this transgenic animal more sensitive. Simple example I'll be giving you. Like I am a mice. They want to test on me the drug of what is cold cough, something you say, cold disease. They want to test on me. I am mice. So what they are doing, directly they are not testing a drug on me. First they will inculcate some gene in my body so that I will be very sensitive to cold. Is it clear? Why they are doing that? Why they are making me more sensitive? Example, coronavirus vaccine they are testing on me. I am a mice. Okay, so first they will make me more sensitive to coronavirus. Why they are doing that? Anyone can you say me? First they will make me more sensitive, then they are using that vaccine, then they will be seeing the result. So why can't directly they can give the vaccine? Why they are making my body more sensitive? Because you will be, the scientist will be getting the result very soon and very quick. Is it clear, everyone? You understood? They are not waiting for many years, many months. Directly they are making their body very sensitive. Afterwards the vaccine will react very rapidly on their body. Understood this one, how transgenic animals are used? So for the vaccine safety means, for using a vaccine, for whether developing the vaccine and all, they are using this transgenic animals. I hope so these 4 points are clear. So moving to the fifth point, chemical safety testing. That is, we need to test the vaccine. What I have discussed, that is also a same thing. Okay, here it is just talking about the drugs, what we are using. Drugs in the sense what the tablets and all we are using. So for that testing also, the toxicity first, the testing of the toxicity is made on the transgenic animals only. Okay, they are exposed to, first I said the transgenic animal. Here you can see this point, just now what I explained, this is here. Transgenic animals can be engineered to carry gene that makes them more sensitive to toxic substances than non-transgenic gene, non-transgenic animal. Okay, first they are making their body more sensitive to toxic substance, then afterwards they are exposed to toxic substance and their responses are studied. I hope so it is clear. Whatever I wanted to explain, it is very clear. Okay, so here what they are doing, they are testing the toxicity substance on them. I hope so you have understood now. So this was the last point of this transgenic animal. So 95% they are using the mice here. Is it clear? So just we will recall here what all we studied. First we studied transgenic animals. So what are they? Okay, so they have their DNA manipulated, okay, modified to express the foreign gene. Okay, so human beings, for human beings they are acting as a model here. Okay, so we can just calculate, we can study, we can see the normal changes in the body function by using this transgenic animal, and we can study different types of diseases, their cures, their effects, their impacts, everything on this transgenic animals. Then comes biological product. So these transgenic animals are not only helping us by they are not only helping us by just showing this impact of the drugs and all, also they are giving us some very important, very useful biological products which are very useful for the humans. Is it clear? So in this, you have seen many diseases, a human protein, alpha-1 antitrypsin, then phenylketonuria, cystic fibrosis, these three diseases we discussed. Then we have studied about the first transgenic cow, that is Rosie. Is it clear? Rosie's milk was very good for the human infants, human babies. This we studied. Then for vaccine safety, before giving a vaccine to a human, it is tested on these transgenic animals. Okay. So polio vaccine is the example which is given here. Then last point we have studied is a chemical safety. First, the body is made sensitive to the toxic substance. Then what happens? They are exposed to toxic substance so that the response are studied very soon. The result we can see very rapid result. Is it clear? So this is a summary. In summary, we can say that transgenic animals are a very valuable tool for scientific research. So whatever the research the scientists are doing, so transgenic animals plays a very vital role in for the humans. They are acting as a models for humans. Is it clear? I hope so this is clear. So now we are moving to the next. So here you can see There are tips for exams, examples, related videos. Okay, so there is one related video to the transgenic animals. So this whatever whatever you are studying in this chapter, it is not like every time you will be feeling like to sit to listen to a lecture. And also sometimes when you are free, when you are having some time, you can just go through the videos also. Okay, sometimes we'll understand clearly by seeing a video. Okay, and these videos are also not so very lengthy that you will getting bored. So these are very short and exact, up to the mark. Okay, what the your chapter contains, what the topic you are having, that only the videos are containing. Is it clear? So whenever you are getting any free leisure time, you can just watch out this video related to transgenic animal. You will be getting a more clear idea to this. Okay, this is very interesting, it is not a boring. Okay, so next you can see there are tips for exams. So before just diving or before just jumping into transgenic animal, first you need to have a clear cut idea about the solid understanding you must have on the concept of recombinant DNA technology, gene cloning, genetic engineering, plasmid, vector gene. These all everything you have studied in the previous chapter, that is biotechnology principles and processes. In that chapter, everything you have studied. So first your basics should be very strong. If your basics are strong, then only you can move to this chapter. Okay, I am saying you have to insert the gene of your choice. Now the question, if you have not gone through the previous chapter, you will be just admiring what is a gene of interest, what is a gene of your choice, so how we must extract, how we must isolate the gene. Is it clear? So everything, for a clearer idea, you must go through the previous chapter, that is the basic of this biotechnology chapter. So by going through that chapter, you will be getting every point in your mind, it will be clear. And you need to know the techniques. Okay, later I will be explaining you. Micro injections are there here, embryonic cell mediated gene transfer, retroviral vectors. So what are the techniques nowadays are taking place, that techniques you must be familiarized with. Okay, so you need to study more and more examples related to transgenic animals. You need to see more and more videos. Is it clear? It is not like by studying some examples you will be getting all the knowledge. So for that purpose, you need to watch some transgenic animal videos. You can watch Rosie video, how they did in very nice animated form, you can see on the YouTube. Is it clear? So like that. Then applications. What are the applications of this? So applications are nothing for agriculture purpose, for medicine purpose. Previously in the for topic 1, first and second topic only, it was about agriculture purpose. Now we are going to study applications of transgenic animals. So what are the applications of transgenic animals? What is they are giving major application is they are giving us by ethical considerations. Then you have to go through the examples, you have to know the techniques, what all is going to happen here. Is it clear? So these are some tips for exam. Now moving on to the examples. Okay, so example number 1, production of human insulin in transgenic animals. So here how the production of human insulin is in transgenic animal, you have to give an example. This is the question. Okay, so in the field of medicine, transgenic animals have been employed to use human proteins. Okay, so here the example is about the production of human insulin. So how human insulin is produced. So for that, we need to choose a objective of our example. The objective here is we must get a human insulin from the goat we are extracting here. Is it clear? From a transgenic goat we are extracting the human insulin. So the objective, produce human insulin in the milk of transgenic goat. So the procedure you can see, first you need to isolate the human insulin gene. So how we can isolate? Again, for that we have to study the biotechnology principle and processes. So in the previous chapter, I explained you the process, that is gel electrophoresis. Just for recollection, I will be explaining you here. Gel electrophoresis. So this, in gel electrophoresis, I have explained you, there will be a, like this, there will be a structure in the comb like structure, plates will be there. So if you remember, I have explained this in the previous chapter. Then some bands will be there like this. Okay. So here what we are doing, we are injecting the DNA in this. So there is a comb like structure when we are putting the DNA in this. How DNA is obtained, that also isolation, you know, I think centrifugation we do, we will add chilled ethanol, what all we are adding to cut down the, what you called RNA, to cut down the protein, protease we are adding. Okay, so everything we are obtaining a DNA, then that DNA we will be adding in this electrophoresis. So we are putting the DNA in them. Then the larger fragments are occupied by the heavy gene and the lighter ones are going down. Is it clear? So afterwards what they are doing, they are removing this comb like structure will be removed. Then we are getting gel bases like this. Then we are getting a tray. Okay, this is a gel, completely gel. Inside one what we are having, this comb like this structure we are removing, we are getting a gel plate. In this we can see some orange color bands will be there. Is it clear? Some orange color bands we are getting. So in them markings will be there. Based according to the markings what we are getting, the gene of our choice, we are getting the desired gene. Then we are not directly taking out, we will be, if I need this one, I will be cutting this portion like this, then I will be heating it, gel will be melted, and I will be left out with the gene of my choice. This is clear? So here we are isolating the human insulin gene. Then we are inserting the gene in a vector. Okay, we have studied about the plasmid also. In a plasmid, it is acting as a vector. So in a vector, we are inserting this gene. Then we are introducing this vector in the goat cell. Is it clear? So select cells that have successfully incorporated the gene. Then breed transgenic goat capable of expressing human insulin in their milk. Okay, so what we have done, we have taken a human insulin gene. All the process we have done, isolation, insertion in a vector, then introducing the vector in a transgenic animal. After inserting, then we will be getting a desired whatever we need. So what we are getting, goat milk can be harvested. After harvesting the goat milk, insulin can be extracted from that goat milk. Is it clear? So here we can get a very eco-friendly, very cost-effective source of insulin we have got from a goat milk. Is it clear, everyone? So previously we have studied the example of a transgenic cow, Rosie. Okay, how we obtained the milk from that cow. Okay, we have mixed two human insulin. Sorry, not human insulin. Human gene we have introduced in the cow, in a transgenic cow. Then we got what we got. Anyone, can you say? Just now I explained what we got. We got rich protein milk in them. Okay, that was very balanced for human infant. Okay, so this was the example, first example. I said there are many examples. You can just go through many examples on the internet also, on the YouTube also. You can go through many examples. Okay, so transgenic mice for cancer research. Again, everything we are doing the same thing. Now here we have taken a mice, and you have to remember, whenever you are studying about the mice, majority of the experiments, majority of the researchers are done on the mice itself. They are showing more familiar features with humans. That's why we are using mice. So mice are more often used in cancer researches to study the effect of specific gene on tumor development. Okay, so for studying this tumor development, for studying this cancer cells, we are using the transgenic mice here. So investigate the role of specific gene in cancer development. So what we need to do, we need to study about a tumor suppressor gene. Okay, for that, with that gene, the target gene which is associated with the cancer, that is needed to insert into a mouse embryo. Okay, so here they are not directly inserting in a adult mice, like how in a cow directly we did, how in a goat directly we did. So here what we are doing, here we are not directly inserting in a adult mouse. Instead, in a mouse embryo we are inserting it. Understand? Why in a mice embryo we are mouse embryo we are inserting that? Because we need to know the changes whenever the mice is developing, because cancer cells are growing very fast, we know. Okay, whenever the development of the mice takes place, so in the development process, what all are the difficulties the mice will go to face, so that all things we need to note down. That's why in a mouse embryo only we are inserting the gene. And this is another different technique, that is micro injection technique. Using a micro injection, a thin needle will be there, so using that micro injection technique we are inserting the gene in the mouse embryo. Then we will be allowing that transgenic mice to develop. When it is going to develop, we must be researching, or we must be studying about the whatever the influence it is happening on the cancer development in the mice. I hope so it's clear. Is it clear? So these were the examples. Now we will be moving on to the questions. So first question, what is the first question? What is the cell that receives a recombinant gene called? So what is that? The cell that receives a recombinant gene is called transgenic cell. You must know. I'll be showing you the answer. See here. The cell that receives a recombinant gene is called a transformed cell, or also we call it as a transgenic cell. So just you say me what the transgenic means. I first. So when are we using this transgenic word? Say me. Answer me. So the term transgenic is used when the genetic material, when the gene from one organism it is transformed to another organism, then only the term transgenic will be coming here. Is it clear? So when we are using that, when genetic material or when gene from one organism it is transferred to another organism. From one organism to another organism, if we are putting a gene, if we are transferring a genetic material, then we use the word transgenic. Is it clear? So in the context of biotechnology, the scientists more often they introduce a recombinant gene. A gene, what is a recombinant gene now? A recombinant gene is a gene that has been artificially created by combining DNA from different sources. I hope so it's clear. In very simpler way I am explaining you. Okay, so what is the recombinant gene? If they are saying in the question only itself they have said na, the cell that receives a recombinant gene. So what is this recombinant gene means? A gene that has been artificially created. Artificially we are creating one gene, and how we are creating that gene? By combining the DNA from different sources. This is a source A, this is a source B. Source A, source B. So source A DNA we are taking plus source B DNA we are taking. Is it clear? When we are combining the DNA from different sources, just now what I said, the DNA, by combining the DNA of different sources, a gene has been artificially created. Okay, artificial gene. We can name it as artificial gene, and this artificial gene is nothing but recombinant gene. I hope so. The recombinant gene is also clear, and the answer, that is transgenic cell, that is also clear. Is it clear, everyone? The first question, if you are getting this question, was asked in the year 2019. Is it clear? What is the cell that receives a recombinant gene called? Now, whenever this question comes, it should be very easy for you to say transgenic cell. Is it clear? Next, we'll be moving on to the question number 2. Why are there no variations seen in clones? State 2 reasons. Okay, so I will be showing you first thing. Okay, clones. Just a second. No issues. I'll be showing you here. So any two reasons they are saying. Okay. So you know first how you can see the answer here. First, I'll show you the answer. See here. No variation in clones because first thing is genetic uniformity. Clones are produced through asexual reproduction. We all know this. Is it clear? So when we are producing through asexual reproduction, this method will involve what? Duplicating the genetic material. It is not dividing the genetic material. First thing you have to remember is duplicating the genetic material of a single parent, that will be leading to offspring. Understood? So that are genetically identical to a parent. Whenever you are dealing with asexual reproduction, the in the options, if you are saying explain what is asexual reproduction, what are the consequences of asexual reproduction, we will be writing one important sentence, that is they are genetically identical to the parent. Is it clear? So no variations are seen here. So first reason why there are no variation in clones, first thing what you need to say, they reproduce through asexual reproduction. Asexual reproduction, this process is involving duplicating the genetic material, and that will lead to genetically identical offspring to their parent. Is it clear? Then second one is mitotic cell division. You know, in a sexual reproduction, mitotic division takes place. If it was meiosis, we could have said that genetic variations have shuffled. But here in mitotic division, it is that form of division which will result in the production of cells with identical genetic material. Is it clear? So mitotic division and genetic uniformity, these are the two reasons why there are no variations in clones. Is it clear? Hello students, I hope so I am audible now. Okay, first let me share my screen. So what we were discussing? We were discussing about the clones, right? So clones are the exact copies of their parents, I said, and they are sharing the same DNA. So they have made without mixing of genetic material from two parents. So that's why they don't have any variations in them. Is it clear? I hope everyone, it is very clear. I have explained in very simpler words here. Okay. Next, moving on to the question number 3. So you can see here third question what it is. What are transgenic animals? Very easy questions I feel, right? Animals with mixed genetic triads, animals from the wild, animals that have had foreign gene introduced in their genome. Got it? Then animals with a single genetic triad. So the answer is transgenic animals are those that have been genetically modified introducing by introducing foreign gene in their genome. That is the answer. That is the option C. Understood? No need to explain because the whole topic we have learned about the transgenic animals only. Okay. So scientists, they can insert the genes from one type of animal or even from a different species also, we can say, into the DNA of another animal. Okay, these added genes will give the animal new triads or new abilities that it would not have normally containing with them. Is it clear what I need to explain? So why they are adding the genes so that the animal will be giving us the new triads or new abilities that it would not normally have. I hope so this is clear. Next, moving to the fourth question, last one. Which technique is commonly used to introduce foreign gene into the genome of transgenic animal? And the option here is micro injections is widely used here. Okay, what is the thing? Micro injections. So mostly we are using micro injections. So now I'll be just explaining you what micro injections they do. So micro injections involves using a very fine needle to inject foreign DNA directly into the nucleus of a fertilized egg cell. Is it clear? I'll be writing it down. It will be more clear. They are using what they are using? Fine needles they are using. By fine needles, what they are doing, where they are injecting, they are directly injecting the foreign DNA into the nucleus of a fertilized egg cell. Nucleus of a fertilized egg cell. Okay, in previous example also, in the mice we have injected using the micro injections, we have injected in the embryo of a mice. Is it clear? Mouse embryo we have injected that. So what is this micro injections mean? They are the fine needle which is injecting foreign DNA directly into the nucleus of a fertilized egg cell. So this process will allow the scientists to precisely insert the desired gene. Okay, this insertion should be very precise. So that's why micro injections, which is having fine needle, I am giving you a pot or something, I am giving you a paper or a cloth. Imagine I am giving you a cloth. Okay, so now I will be saying you, this is a piece of a cloth I will give you. Example I am giving. Now I am giving you this thickness pen. Okay, you can see the thickness of my pen. This much thickness pen I will be giving you, and I will be saying you mark at this point. The pen's needle I am saying, not this much thin, this much big needle it will be having. I will be saying you to point at this point. So what you will be doing, you will be pointing at this whole area, because the thickness of the pen is this much. Okay, no precise pointing will be there. So instead of this, if I am giving you this thickness, the short, like very thin thickness needle I am giving you, then if I am saying you point out to the point, then you can easily point out the point which I am saying. Understood what I am going, what I am explaining? So because of using that thin needle only, precisely the scientist, they can insert their desired gene in the genetic material of the developing embryo. Is it clear? I hope so you understood. So how it works means, the scientists, what they do, they first isolate the gene of interest. Okay, how to isolate and all, you know. The first they will be isolating the gene of interest, and then they prepare it for injection. Afterwards, after they will be having the injection, they carefully, what they do, they inject that in the, where, pronucleus of a fertilized egg. When the egg is fertilized, in the nucleus of the egg, they will be inserting this injection. Typically it we call it as a, what you call, just, yeah, single cell stage. In a single cell stage, in after fertilization of an egg, in the nucleus, that will be injected. Afterward, as the embryo continues to develop, the foreign gene will integrate into its genome. Okay, afterwards when we are adding that, you cannot see the results. Okay, so previously only in the nucleus only they are adding this gene, so that when the embryo development starts and when the embryo development goes on, the foreign gene should integrate into its genome, and the resulting animal, if the process is successful, then it will be carrying the inserted gene in all its cell. Okay, if it is inculcating it in all its cells, means then we can say the process was successful.